Hello and welcome to Life Sciences, A Day in Our Lives. I'm Annie and I'm a PhD student at the University of Cambridge. I've got together a group of PhD students who are all doing life sciences research right now, so you can hear straight from them what it's like. They've got under 3 minutes to give you a glimpse into their daily life and how they ended up where they are now. Thanks for joining us, and if you have any questions for them, please put them in the YouTube chat or you can tweet them after the event as well. Some of our PhD students will be returning for our sister event, which is called A Young Person's Guide to the Life Sciences. If you want to hear a bit more in depth about what doing a PhD is like and hear us discuss some big questions about life sciences research, then please check it out. Without further ado, let's get started.
My name is Tapoka and I'm a PhD student in parasitology at the Wellcome Institute and the University of Cambridge. The parasite that I study, or that I'm interested in, is called the whipworm. Whipworms are roundworms or nematodes that infect over 400 million people across the tropics. The whipworm life cycle begins when people eat food and water contaminated with the parasite eggs. These eggs then go into your intestine where they hatch, the worms swim out and burrow into the intestinal lining, causing an infection. This hatching process comes as a result of the parasite eggs talking to the bacteria that live in our guts. I'm really interested in understanding this communication between parasites and bacteria and how we can use that to help people get better. My day is either in the lab or at my computer. When I'm in the lab, I spend a lot of time looking down the microscope, looking for hatching events, and I grow lots of different types of bacteria from all sorts of places, and I use these bacteria to try and make my worm eggs hatch. When I'm working on the computer, I personally look at genomics, which is DNA, transcriptomics, which is RNA, and proteomics, which is proteins, just to try and get an understanding of the proteins and molecules that are in the whipworms and the proteins and molecules that are in the bacteria that cause hatching. Because the gut is such a diverse, busy environment, with lots of different organisms, even just studying gut bacteria, talking to one type of worm, means I often end up with more questions than when I started. But having this whole load of questions is part of the reason why I love research. It helps me satisfy that curiosity I have about understanding how the world works and getting to know what makes life tick. I sometimes like to think of myself being a scientist as being a bit of a detective, armed with a microscope instead of a magnifying glass, trying to solve the mystery of life as we know it.
Hi, my name is Jascha Achterberg and I'm a PhD student at the MRC in Cognition and Brain Sciences unit here in Cambridge. I do computational neuroscience and I'm specifically interested in how does intelligence happen in brains and in machines. Basically, what you have to imagine is your brain is a huge network of cells, and those are sending signals back and forth for the entire day, all day long. And somehow this endless pinging back and forth of signals in the brain gives you and humans in general an absolutely astonishing ability for complex thought. Specifically, I look at brain regions in the frontal part of the brain because those are especially associated with kind of complex thought and complex problem solving, giving you the ability to plan ahead in like a very detailed way, but also humans in general to solve complex equations which are needed to build rockets to fly to the moon. What my day-to-day life is like is I get a huge amount of data, of mysterious brain data from those kind of frontal regions, and have to somehow make sense of it. I sit down and face this kind of creative challenge of which mechanisms can I invent and come up with which could possibly happen in the brain and could then explain the signals that we observe. Often we kind of design theoretical models or the theoretical processes, implement them in the computer, kind of like, like a mini artificial brain, which has different parameters and different characteristics, which we also assume to be true for the real brain. And then we see whether this mini artificial brain comes up with kind of the same behavior or same internal signals as the brains that we observe in real life. quite often you get stuck. And whenever this happens, you talk to your friends and colleagues in either the same kind of scientific discipline or completely different discipline. Talk to them about your scientific problem and generate a dialogue. Because one of the things really is that in those kind of complex problems in science, there's no way of doing this alone. And this is really what makes science so fun, in my opinion. It really is a collective challenge, making progress step by step and trying to understand how nature works together.
I love it.
Is it a primary? Oh. So yeah, I was trying to do more still LED here.
Hi, my name is Tian. I'm a medical doctor who recently finished a PhD in clinical research. My interest is in developing new medication for patients who have heart attacks. What I did for my PhD was recruit patients who were admitted as emergency with a heart attack and give them an experimental drug. We then follow them up for over 6 months and to see how they get on, and to firstly see if the drug was safe and to see if there are any signs that the drug was effective.
Consenting people for clinical trials goes something a bit like this.
Oh, hi John. Hi, my name is Tian. I'm the heart doctor here involved in research. I'm sorry to hear you had a heart attack. We've managed to treat your heart vessel and open it back up, but unfortunately there's been some heart damage. One of the things we're doing here is looking at whether we can improve that heart damage using experimental drugs. Well, we're running a clinical trial here. Would you like to learn more about it and potentially take part in a clinical trial?
Yeah, sure.
Okay, great. Well, I'll leave you some information and you can have a read about it.
Thanks for joining me today. Goodbye.
Hi, I'm Annabel. I work at the LMB, and today I'll be showing you an average day in the life of a PhD student. My work looks at fruit fly embryos, specifically what genes are expressed in a small patch of cells that will eventually form tubes. I want to know what genes are in this patch of cells and what those genes are doing. In my project, I frequently use a technique called in situ hybridization to find out where a gene is expressed in embryos. You can think of this experiment a bit like making a glitter painting. First, you add glue. The glue I use are small pieces of RNA which will only stick when the gene I'm looking for is being expressed. Then you remove the glue from places where it hasn't stuck, or in my case where the gene isn't being expressed. Then you add glitter. In my case, this is a probe with a fluorophore attached, and this allows me to visualise where my probes have bound and where my gene is expressed. We let that sit for a quick lunch break, and then I do one more wash to make sure the signal is specific, and then we can mount the embryos ready for the microscope.
No! No! No!
On the microscope, I can start to choose which embryos I want to image. I choose embryos based on the point of development they're in and what position they're in. Once I have my images, I can post-process them to make sure the images are crisp and bright. And without further ado, here is one of the images I took today. Here are the two patches of cells from the glitter painting example. I also use other techniques which can outline the cells and also to tag where the nuclei are. I can put them all into the same image, and I'm left with a pretty nice image of when and where this particular gene is expressed. One of the aims of my project is to build up a database of different genes to map which genes are expressed and where right before tubes begin to form. And if I'm lucky, some days I get to look at genes which don't have official names yet. That means I might be the first person in the world to know where these genes are expressed in the embryo, and if I'm even luckier and that gene does something cool, I might be able to name it in the future. If you were allowed to name a gene, what would you call it? In the fly world, people have got pretty creative with it. Thank you so much for tagging along today. I hope you learnt something, and now I have to go and clean up all that glitter.
Hello, my name's Henry, and I'm a PhD student at the University of Cambridge, where I study viruses and how viruses are able to spread from cell to cell. My journey to a PhD started when I was growing up and when I was at school, where I had this interest in how things worked and in the science of the world around me. I had a particular interest in the natural world, ranging from animals and plants right the way through to infectious disease. However, when I finished school, I didn't know that I wanted to do research and do a PhD, and actually I wanted to be a vet. But I didn't get into veterinary school, and so ended up studying biology at university. It was during this time at university that I gained this real interest in infectious disease and realised that I wanted to pursue research into a PhD. I wanted to ask and answer questions that genuinely nobody had ever answered before. And for me, it's this process of discovery which makes a PhD in research so appealing. So, as I mentioned, my research is on viruses and how viruses are able to spread from cell to cell. To study this, I'm able to grow a variety of different cells in the lab and infect these cells with a variety of different viruses. I can then observe how the virus spreads and observe changes in the cells under different conditions. I use different techniques and a variety of techniques to study this interaction between the virus and the cell. For example, I can use powerful microscopes to look at how cells change when they get infected. Now, I do spend a lot of time in the lab doing experiments, but lab work is only one part of doing a PhD in the life sciences. My days can also involve me meeting with my supervisor and other researchers, reading, analysing my data, making presentations and presenting my work, as well as listening to other people present their research. All of this means that no day is ever the same, and there's always something to keep you busy and engaged.
Hi, I'm Julia, and I'm a PhD student at the MRC Epidemiology unit. During my PhD, my research has mainly focused on improving our understanding on how and why some people develop type 2 diabetes. Diabetes is a disease in which the body cannot properly use glucose, and it manifests itself as high blood sugar levels. How do you actually study these in humans? We usually do this by taking a group of people and studying them over many years, after which some will develop diabetes. Our goal is then to identify which characteristics were initially different in the people who got diabetes compared to those who didn't. What sort of characteristics do you study in these people? Well, we study a range of things, including their age, sex, their body weight, and we even go down to a molecular level, and we can study differences in their genes and molecules that are found circulating in the bloodstream, such as proteins or metabolites. I work on analyzing all of these people's characteristics, and because we can have millions of characteristics for thousands of individuals, this information is simply too much for humans to analyze, and we need the help from very powerful computers to help us do it. How powerful are these computers? Imagine combining thousands of normal desktop computers together. That's how powerful we're talking. My typical day involves me writing code on these supercomputers to process all of this information. You may have seen examples of code looking something like this, which may look really scary and make people think you're a hacker, but in reality, it's just a set of instructions to tell the computer what to do with their data and how to analyze it. I see. But why do you want to understand what was different initially in the people who got diabetes compared to those who didn't? Ultimately, if we can better understand this, we can then have a look at any other individual and make a better guess on whether he or she is at very high risk of developing diabetes in the future. This can then help on improve the way we treat this disease from the very early stages and hopefully delay or even prevent people from getting it. Cool. Can you only do this for type 2 diabetes? Not really. Although during my PhD my main focus has been on diabetes, this strategy is actually widely used to study many other diseases. This can then improve our understanding on the underlying reasons on why some people may be more prone to develop certain types of diseases.
Hello, my name is Simpson-Kent, and I am currently a fourth-year PhD student in medical science at the University of Cambridge, where I study developmental cognitive neuroscience, which basically is I study how cognition evolves in the brain, especially from childhood to early adulthood, so about ages 5 to say about 22. And basically, so my day to day, I don't really have any. I don't really work in a traditional lab, so to speak. it's all just doing coding and computer coding and data analysis. So data analysis is sort of similar to this great book called Brain Network Analysis, which basically looks at the brain as a network of interacting elements that together produce cognitive ability, or what we call general intelligence. And so I study how this develops in the brain, hopefully so that I can then apply this knowledge to education research to help kids, particularly those who are struggling and from disadvantaged backgrounds, learn better.
Hi everyone, my name is Matt Keeble. I'm a second year PhD student based at the Centre for Diet and Activity Research within the University of Cambridge. Ever since I was young, I was interested in how the human body works, and especially how what we eat can influence how we feel, our waistlines, and eventually our health. I first got into research when I left school to study a sports science degree. I thought that food was just about performance, but actually, I realised that it can influence things at a population level. So I then went and studied a second degree in public health nutrition. It was when I was studying this degree that I got really interested in the food environment and how everything around us can influence the different types of food that we access, what we decide to eat, and what that might mean for our health. When I finished that degree, I moved to the University of Cambridge, where I started to do research for other people. That was great, but I knew that I wanted to do my own research and answer my own research questions. So I managed to secure some funding for a PhD. And now I'm researching online food delivery services. I'm interested in how they influence the types of food that people eat, what that might mean for how often they eat takeaway food, and what that might mean for their diet and their health. My days never look the same. Some days I'm speaking with online food delivery service customers to try and find out what they like about these services and why they use them. Other days I'm mapping online food delivery service food outlets to try and find out which areas in the UK can access the most or the least and why that might differ. Other days I'm analysing data that's already been collected to try and find trends in online food delivery service use and how that might influence people's health. I think that's what I like most about research, is that my days are never the same. In the future, I want to carry on researching online food delivery services and what that might mean for health in other countries where these services are only just becoming established. I think that research is a great career, and I would highly recommend it if you're interested in finding out more about specific topics and researching them in real depth. Thanks for listening.
Welcome back. We've reached the final diary entry, and that's actually from me. So as I said, I'm a PhD student and I'm doing this at the MRC Cancer Unit, where I'm researching how and why cancer develops. This is a huge question, and it actually encompasses so many different aspects of cell biology, and that's partly why I got so interested in this area of research in the beginning. Specifically, I'm looking at the conditions required for a cancer to develop, and I'm looking in particular at cell metabolism, so what the cells are eating and what they're doing, and this can affect how they behave. So cells are always making what we call decisions, so they're either deciding to live, to die, to move around, to replicate, to make more of them. And cancer cells make the wrong decisions in these contexts. But why is a little bit unclear, and I'm trying to understand how metabolism can affect these decisions that they're making. Unlike some of the other PhD students you've heard from, I fit the more traditional mould of wearing a lab coat and pipetting a lot of the time. I'm trying to develop new methods so that we can look at metabolism one cell at a time. And we use a certain type of microscopy where we've edited the cancer cells so that they emit a certain type of light, they fluoresce, and we can measure that light. Now what we've done is edited the cells so that the light they emit changes based on the concentration of a certain metabolite that we're interested in. I'm mainly looking at ATP. And by that way, we can look at the metabolism, each cell, one by one. This essentially means I spend a lot of time in dark rooms looking at the cells emitting their little lights. But I also do some experiments in the light where I look at how the cells are signaling, so how they're reacting to the environment around them and to what's going on in the cell itself and their neighbours. I also look at how they are metabolizing as a whole population. And I spend a lot of time looking after the cells and editing them so that they can glow in this certain way. We can use all of this information to look at cancer at its very beginning and try and understand why a tumour develops and ultimately help us treat it a bit better. I work with two great teams of people. One is a lab that focuses on this certain type of microscopy. One is a lab that works on cancer metabolism, and together we're working to address this problem. So I spend a lot of time with them doing experiments, discussing my data, presenting, and it's definitely a team sport. That was a lot of information in a short amount of time, but I hope you're still with us. I hope you've had a bit of an insight into what doing life sciences research is like and some of the amazing research that our PhD students are doing. Thank you so much to all of our speakers, and thank you so much to you for joining us. If you have any questions, please put them in the chat or tweet us now. And don't forget to check out our other event, A Young Person's Guide to the Life Sciences.